Hello, everyone, and welcome to today's presentation titled High Quality GPCR Antibody Discovery Using Full-Length Nanodisc GPCR Proteins and Single B-Cell Technology. This webinar is part of the 9th Annual Drug Discovery and Development Virtual Event Series. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by DIMA Biotechnology. For more information about DIMA Biotechnology, go to www.dimabio.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate first by communicating with other attendees via our live chat feature during the presentation. You can find the live chat located on the left side of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type those questions into the Ask a Question box and click Submit. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Donghui Ma, PhD, CEO, DIMA Biotechnology Ltd. For a complete biography on our speaker, please visit the Presenter tab in the menu on the left-hand side of your screen. Dr. Ma, you may now begin your presentation.
Yeah, hello everybody. This is Donghui Ma from DIMA Biotech. Today I'm going to present technology from DIMA Biotech on Nanodisc technology and its application in therapeutic antibody discovery. And DIMA Biotech is a biotech company focusing on head to lead stage of antibody drug discovery. We mostly focus on the preclinical research for the therapeutic antibody development. In today's talk, I will divide my talk into 3 sessions. The first part, I will talk about DIMA's development discovery platforms for therapeutic antibodies, from immunogen preparation, antibody screening, and after identify the lead molecule, we'll do like humanization for antibody engineering and also affinity maturation. The second part of the talk, we will showcase 2 of the examples of multipass transmembrane protein, how we develop like a loop-specific antibody, and also from a lead to IIT clinical stage, and in another case is GPCR targets. On the third part, we'll showcase how we help the biopharma company to speed up their discovery and process to push more candidate into clinical trials, and our special like offer off-the-shelf lead antibody molecules. And so the first part I'll talk about our transmembrane protein technology platform, which utilized for the therapeutic antibody discovery. As everybody know, for therapeutic antibody, they are functional monoclonal antibody, have to work in the 37-degree human body and to coordinate like an immune system to attack like a cancer cell in the case of oncology. And the most important thing to develop a functional therapeutic antibody, the functional protein utilized for immunogen or screening is very critical. People say garbage in, garbage out. And currently for therapeutic and monoclonal antibody, more than 90% of them are membrane protein as a target. To group them into the, based on the complexity of their transmembrane part, we can divide them into 2 categories, single transmembrane protein and a multi-span transmembrane protein. So I show on the right side. And for the single transmembrane protein, people can directly cut the extracellular domain, make a soluble protein as an immunogen or screening protein. There's no problem. A lot of FDA-approved drugs developed by this approach. For multipass transmembrane protein like a GPCR, as shown in this figure, If you just cut the loop region and the N-terminal region, they are very hard to mimic the native conformation because the loop region has to coordinate with the transmembrane part from the like a tertiary structure. So in this case, we currently DIMA has a different technology to make native protein. One is use a non-purified vesicle format, so the protein still keep in the membrane vesicle format. Another is a purified form. So today I will be talking more on the ECD fusion proteins, synthetic nanodisc, PeptiNanodisc technology. And the first, I'll talk about the ECD, traditional people use for single transmembrane protein. And for the ECD fusion protein and functional evaluation, what we do is all this protein we purify by HK293 cell, mammalian cell production and also purification. And to ensure the protein in the native conformation and functional, so if they do have a receptor or ligand, we'll synthesize the receptor ligand to ensure they can bind in the in vitro status. If they don't have the corresponding counterpart of a ligand or receptor, we'll see whether there's a benchmark antibody in clinical trial or FDA approved drug, then we will synthesize those to bind to ensure the binding towards the protein we use for immunization and screening. So that's then we'll jump to the multipass transmembrane protein, talk about our DIMPro technology platform. For the multipass transmembrane protein, one of the major challenges is there's a lot of challenge for this. So one of this it is that they are highly hydrophobic because you want to keep the transmembrane protein, transmembrane part intact. So they are highly hydrophobic. They tend to form aggregate. So when you purify them in the aqueous solution, they will lose their function and the conformation. And so far, there are more than 300 druggable targets for GPCR, but right now only 3 of them get FDA approved, therapeutic antibody. So one of the major challenge for this, it is not the antibody discovery part is tough. It is the immunogen functional protein preparation is very hard to make. That's why it's very hard to make all those therapeutic for GPCR antibodies. At DIMA, we try to solve this issue by using our different technology platform. So first, I will talk about non-purified membrane vesicle format when we utilize to make these full-length membrane proteins. So right now, we offer 3 different technology solutions. The first, it is a membrane nanoparticle solution. This is developed by DIMA Biotech. Basically, we overexpress the protein on the cell surface. Then we break the cell and isolate the membrane and make them as small nanoparticles and use those for the immunization or screening. The second technology we also currently offer, it is virus-like particle, VLPs. So basically, we utilize the special function of lentivirus GAG protein. They can self-assemble as a vesicle bodied out from overexpression cell, and then they carry out the overexpression multipass transmembrane. And the exosome is similar, but it is a natural process of like a bodying of a nanoparticle from cell surface, and they carry all these multipass transmembrane proteins. For the vesicle approach, one of the major problems is they are not purified. So not only contain the overexpressed multipass membrane protein, but also contain large amount of non-relevant cell surface or membrane scaffold proteins. So it's very hard to quantify the total true amount of target protein. And the second, it is the production. It is highly variable from batch to batch because it depends on the physiological status of the production cell line. So according to our past experience, they are not suitable used as an immunogen alone. So sometimes it's good to combine with nanodiscs or other purified protein as an immunogen or screen, and also screening also has some interference. And so let's jump to our technology on purified multipass transmembrane protein technology in DIMA Biotech. So right now we offer 4 different solutions. First is we call the synthetic nanodisc, and the second is the MSP nanodisc, which is a traditional nanodisc technology. People use ApoA1 protein to form the nanodisc. And the non-lipid platform, we use a PeptiNanodisc and a detergent, traditional detergent technology for purified multipass transmembrane protein. So today I will be more focused on the synthetic nanodisc and the PeptiNanodiscs. And so in general, we always call the synthetic nanodisc called a nanodisc in the forward slides. So nanodisc initially developed by University Professor Steven Snigar's group in 2002. So basically, they use purified ApoA1 protein and assemble with multipass protein and artificial lipid from as a disc-like structure. So this disc-like structure is very small, 5 to 15 nanometer in diameter. So they are totally water aqueous soluble in the solution, so they can be purified to high homogeneity through chromatography. On the right side of the corner, it is a traditional detergent base. So comparing to detergent base, there's quite some advantage comparing to the detergent base. You can see by using detergent, they remove the lipid bilayer, and the multipass protein, they are not reside in the native status, because sometimes the lipid bilayer, the lipid molecule help the functionality of the multipass protein. So in detergent approach, you remove the lipid bilayer, they wrap around the hydrophobic transmembrane part by the amphipathic molecule of the detergent. Sometimes the protein may not exactly recapitulate the functional property of the multipass protein by detergent. So we also did some comparison studies to see how is the purification, the purity status by using detergent, PeptiNanodisc, and the nanodisc technology. From this slide, you can see just one round of affinity purification. Detergent approach, you tend to carry a lot of co-purified hydrophobic protein, which are not present, not quite pure. But by using nanodisc, synthetic nanodisc, and PeptiNanodisc, you can see the purity is pretty good. And in this case, we use a Claudin 18.2, and it's a protein around 27, 28 kilodalton, and they form a dimer and looks like a trimer status. And you see 3-band, but we also showed by the Western. Yeah. And also, our synthetic nanodisc comparing to MSP-based nanodisc, there's another advantage. It is since we don't use scaffold MSP ApoA1 protein, you can see on the right side. So we only have our target protein is in a high homogeneity on the left side. You can see this Claudin 6. You see only 1 band. They are aqueous soluble, and you can do all different assay, ELISA or some other assays. And on the right side is a traditional MSP-based nanodisc. You see a large amount of MSP and also your target protein. But in this case, if you use for immunization, you generate a lot of non-specific antibodies. And also, if you do screening, you tend to interfere. This MSP might interfere with your downstream screening. And Nanodisc can be utilized for different applications, for antibody binding kinetics analysis, phage screening, antibody screening, or small molecule drug screening. You can mimic the native conformation and the protein-lipid interaction, or most recently people use for cryo-EM study, or even drugs delivery. So here we showcase one example. We use our purified 18.2 nanodisc, so we can measure the binding affinity with this rituximab. It is a Stella approved antibody drug for 18-Claudin 18.2. In the past, it's very hard to do this assay, because you don't have purified full-length protein to coat on the chip. So with this approach, we can perfectly measure the binding affinity. And also, PeptiNanodisc, like synthetic nanodiscs, sometimes they can be utilized for different assays. So synthetic nanodiscs is not good for cell-based assay because they carry a patch of the membrane. When you incubate with cell, they sometimes tend to non-specifically fuse with target cell, so you get a background. But PeptiNanodisc, they don't have this problem. So here is the example. We use PeptiNanodisc for Claudin 18.2 CAR T cell, like transduction efficiency measurement. So in this case, it works perfect. And also sometimes synthetic nanodisc and PeptiNanodisc, even on a single target, they might have potential influence on the target protein conformation. So this is a CD19 protein, and we develop both synthetic nanodisc and PeptiNanodisc. We measure the functional property by different therapeutic antibody, and it shows PeptiNanodisc works perfectly in the ELISA, and synthetic nanodisc doesn't work. But on other case, it works in the synthetic nanodisc. And currently, DIMA offer the largest collection and globally for the nanodisc products off the shelf. So this is the comparison of our company with the competitors. And so DIMA is a therapeutic antibody discovery company. Even though we focus on the antibody discovery, we put a lot of effort on the functional protein preparation because we truly believe without a good functional protein, the downstream screening it will waste of time. So that's why we have all different platforms for the multipass functional proteins. Right now, I will jump to our single B cell discovery platform for the antibody discovery. So this is a development process for the single B cell monoclonal antibody discovery platform, or DIMA platform, and we immunize the animal. Then we isolate the PBMC or like a spinosite. Then we use protein to enrich these B cells. After we disseminate this B cell into 96-well plate, and we use our DIMA's proprietary B cell culture media, and let this memory B cell to cultivate in this culture media. So during like 9 to 10 days, this single B cell will proliferate from 1 cell to 100 to 200 cells, and also convert this memory B potentially to plasma cell and a secreted antibody, so we can do assay to identify positive clones and clone the gene. And also we do have like a VHH phage display technology for VHH antibody development. So here's the development process. I won't say more in detail. And so after identify the lead molecule, so those animal derived antibody you can't use for human therapeutic purpose. So DIMA has this DiLibrary antibody engineering platform. We use mammalian cell display technology to do humanization, maturation. This diagram shows how we utilize this platform to display antibody on the cell surface. So basically, we add a C-terminal transmembrane portion to the antibody, and also add an EGFP to track the cell surface expression level. So by this approach, so you can display the whole antibody on the cell surface, and also can monitor and also use target protein to enrich this cell to identify the cell display with high affinity binders. So the reason why we choose mammalian cell display technology instead of traditional phage or ether display, because the mammalian cell has a very good internal quality control system for those mammalian protein and for the antibody. So it will be provide a seamless technology transfer to the downstream CMC, because in the CMC manufacture, people mostly use CHO cell for the antibody production. So by using mammalian cell display platform, we can leverage the internal quality system. If the antibody has any trivial defects in their like a structure, so they will be cleaved off by proteasome or lysosome quality system to get rid of them. So they won't be able to show good cell surface expression and the good binding. So in this case, the molecule we screened will be have a better developability potential. So in the next couple of slides, we will showcase two examples of utilizing DIMA's Nanodisc technology for the mAb discovery. In the first case, I will showcase how we utilize DIMA's second nanodisc, PeptiNanodisc technology to screen loop-specific monoclonal antibody, especially for, in this case, is ECL1, extracellular loop 1. So Claudin 18.2 is an gene is like a specifically overexpressed or like exposed, this protein exposed in the gastric or pancreatic tumors. And in the normal case, they also express in the junction part. They are not exposed for the antibody. So when in the tumor case, they will overexpress and expose. And also there's an isoform for Claudin 18.2 is 18.1. So these two genes come from the same, these two proteins come from the same gene through alternative splicing. The only difference on the right side, you can see the only difference from 18.2 and 18.1 is their extracellular loop 1 on the extracellular side. So how to really develop an antibody only recognize ECL1, but not ECL1 for the Claudin 18.2, not 18.1 is a pretty challenge, because you have to develop an antibody only recognize 18.2. So you only have one choice of develop an antibody against ECL1. And the difference only 9 amino acid. So in this case, we start generate functional proteins and a cell line for the immunization and the screening. So we develop a K562 cell line overexpress Claudin 18.2, and also we made second nanodisc, PeptiNanodisc for the 18.2, and also we made 18.1 PeptiNanodisc for the downstream screening. And so during the initial immunization schedule, you can see initially we used a stable cell line, so they didn't generate a good enough serum titer. Then we start to boost them with nanodisc, purified nanodisc, like 18.2. After immediately after boost with nanodisc, the serum titer get boost up. And so over the course, we constantly, we did several rounds of nanodisc immunization. And so this is the screening approach. We utilized the PeptiNanodisc for 18.2 to do the depletion. So any cross-react with the Claudin 18.1, we use PeptiNanodisc 18.1 to get rid of them. The remaining B cells, then we go through another column of the Bs with a Claudin 18.2 PeptiNanodisc. So only those specifically recognized Claudin 18.2 B cell will be enriched. Those cross-react with 18.1 will be get rid of in the first round of PeptiNanodisc for 18.1. So after we disseminate in this 18.2 specific B cell into 90 plate and clone them, and we successfully got a couple of clones. Here is the functional evaluation. So all these 18.2 specific clones, we made them as a CAR T cell, and so we test with K562 Claudin 18.2 target cell, and also the control we use Claudin 18.1 transfected stable cell line K562. So from this CAR T tumor CAR T cell killing test, you can see our 18.2 specific mAb converted CAR T cell shows a very specific to cell killing effects. And BMK, we like a benchmark, we use CT041. This is a benchmark in clinical trial. These are 4 of our clones showing comparable functionality and comparing to the benchmark antibody and also specificity. So that shows how we can utilize PeptiNanodisc and the synthetic PeptiNanodisc and also synthetic nanodisc and the PeptiNanodisc in terms of the immunogen preparation and also screening for transmembrane protein. And so in the next case, we'll show a GPCR target. We show how we work with a CAR T company. So they do the CMC manufacture and also clinical trial, how we from the lead discovery to IIT clinical trial. So this target we choose a GPCR target called the GPRC5D is a druggable target for multiple myeloma. So currently, there's quite a number of different modalities in clinical trials, including CAR T, dual CAR T, and bispecific antibody and trispecific antibodies. So right now, J&J's mAb is already approved for the it's a bispecific antibody already approved by FDA for the multiple myeloma treatment. To develop this antibody is quite a challenge because this GPCR target have a very small loop, and also the intermediate part only 27 amino acid. How to develop this antibody is a challenge. So currently, we already developed 9 humanized CAR construct, and the antibody convert to the CAR construct. We also explore other potential drug modality for this group molecule. So after we so we are trying to make CAR T construct. So we first convert this mAb into SF3 format. Then we did a flow evaluation. First with native cell line RPMI8226, this cell line, they express GPRC5D, and we did a flow test. It shows several of our candidate molecule shows a very good binding affinity in flow comparing to the benchmark currently in clinical trial MCAR-H109. It is BMS, CAR T in phase 2 clinical trial. And on the right side is with a stable cell line. And after we out of this 9 molecules, so we identify 3 of them to proceed further into animal in vivo assay. So in this G05, 07, and 09 candidate, we made a CAR T cell and infuse into xenograft animals. You can see here, all three of them show pretty good tumor suppression. And to choose one of them for a clinical trial, so we also test target specificity by membrane protein array. Our G07 show very good specificity, bind only in this array only to GPRC5D and another protein which later when we do further tests it doesn't bind, and it also is mainly located in the ER. And currently we work with a Chinese cancer hospital. We by April of last year, we already infused 18 patients, and they all shows very good clinical effects. Our CR rate is about 65%. And this is phase 2 clinical data from BMS, and they did more than 100 patients, so their CR rate is around 48% of their optimized dosage. And so here is a summary of DIMA's products we offer to the biopharm customers for preclinical research need. So far, we have 2 of our flagship products. Right now, we are global #1 in terms of the collection. First is a full-length multipass transmembrane protein in Nanodisc format. Second is our off-the-shelf lead molecules. So we already developed more than 500 druggable targets with more than 5,000 validated therapeutic antibody, and we did a mostly in vitro assay. So they are off the shelf. So biopharma company, they can get the molecule next day for internal evaluation. And so on the third part, I will here to show very quickly about our business model, how we help biopharm for their drug discovery at the preclinical stage. So all these 5,000 molecules, they are already pre-validated with a data package and off the shelf, so the biopharm customers can contact us. So we can we sign the MTA, we send you the data package. You can take a look and see whether fulfill your needs. Then you can start the test next day. So we guarantee no upfront payment, no risk, and no waiting time. So we really can expedite your discovery process. Thank you. Thanks for your attention. I would take questions.
Thank you again, Dr. Ma, for your presentation. And we will now begin the live Q&A portion of the webinar. So if you have any questions that you'd like to ask, please do so now. You can do that by submitting the question in the Ask a Question box, which is located at the bottom of your screen. We will answer as many questions as we have time for today. So let's go ahead and get started. Our first question asks, how does SPR work in practice with indigenous proteins using synthetic nanodiscs in terms of immobilization and ensuring correct protein orientation?
Yeah, so basically, whether it's endogenous or overexpressed protein, can both work with the SPR technology to measure for the small molecule binding or antibody binding affinity. And in terms of immobilization, there's multiple different ways. So people can do like a mean immobilization, or we can add a AVI tag and biotinate it, and so those can be also biotinated chip to immobilize the protein. And in terms of the ensure the correct protein orientation, and with biotin immobilization with the AVI technology will be much ensured. Yeah.
Wonderful. Thank you so much. Our next question, MSP nanodisc size is controlled. What about the size of synthetic nanodiscs?
So MSP nanodisc, according to the publication, it might be more well controlled. And for synthetic nanodisc, ours is more SMA based. So according to the past publication, the size might be variable. Yeah, so it's not as well as controlled as MSP. Yeah.
Wonderful. Thank you for that, Dr. Ma. Our next question, which platform is being used for B cell enrichment and single cell well dispense? Is any additional enrichment performed with nanodiscs or otherwise prior to sending hits to sequencing?
We basically use FACS sorting for the B cell enrichment, or sometimes we use max enrichment. I mean, to enrich, we immobilize the target protein on the magnetic beads and then use the magnetic beads to enrich the memory B cells. Then we cultivate, and after we cultivate for like 7 to 9 days, after identify the positive clones, and those cells will also will proliferate from 1 cell to 100 to 200 cells. Then we send those cell for cloning and sequencing. Yeah.
Wonderful. Thank you. Our next question, what kind of storage condition is recommended for nanodisc and PeptiNanodisc?
According to our experience, the nanodisc and PeptiNanodisc is pretty stable. And the nanodisc, synthetic nanodisc, is even more stable on the regular, like a 4-degree condition, or even room temperature is pretty stable. We tested in the past. We put it into room a temperature. They are pretty stable actually. So when we ship all those products, they were in lyophilized format. So you can store them for long-term storage in -80. And after add water to reconstitute into aqueous solution, we recommend you to store at a 4-degree. Yeah.
Wonderful. Thank you so much. Our next question asks, what is the final product formulation for nanodisc and PeptiNanodisc products?
And for the final formulation, we lyophilize in the trace containing buffer, and also contain 5 to 8% of taholos, and so that's the final formulation. Yeah.
All right, we're getting a lot of great questions. Please continue sending your questions through the Ask a Question box here in our virtual studio. Our next question asks, will DIMA provide development service if my target protein of interest is not available as an off-the-shelf product?
Yes, we do provide service. So if your interest target is when you search on our website, you don't see your interest target, you can always submit the service request to our info at dimabio.com and website. So we can do customer service for the production. Yeah.
All right, thank you so much. Our next question, what are the major differences between synthetic nanodisc and PeptiNanodisc?
The major difference, synthetic nanodiscs contain a membrane patch in the center, so it will circulate by the synthetic polymer, so still have the original membrane patch with the protein. So the PeptiNanodisc initially solubilized by the detergent, then reconstituted with a peptide, and so they don't contain any membrane patch. So the good thing with PeptiNanodisc, can be utilized for the live cell, like for the live cell imaging, or you can use for the cell-based assay. For the synthetic nanodisc with the membrane patch, they tend to non-specifically fuse with the cell, so they create a lot of background. So I would not suggest you do a cell-based assay with the synthetic nanodisc. I would suggest you do with the PeptiNanodisc. But synthetic nanodisc you can use for like protein-protein interaction and small molecule protein interaction measurement. Yeah.
Perfect. Thank you so much. And I think we have time for one or two more questions. So Dr. Ma, if you'll look at 12 and 13 and just let me know in chat if those will work. We do have another one that I know we've already approved. And that question is, for PeptiNanodisc, what are the recommended applications?
The recommended application for PeptiNanodisc, as I just mentioned, it can be used for the cell-based assay, and you can use that for the like a cryo-EM, and so that's, and there's a lot of other things you can do, and also you can use for like as an immunology, for antibody generation or the screening, and so that's what we can. I think there's one question in the group chat box. You mentioned SMA-based synthetic nanodisc. Do you experience any UV interference issue in your workflow? If so, how do you overcome that to determine the accurate protein concentration? Yeah, that's a good question. So yeah, the SMA-based polymer, they will interfere with UV measurement. So normally we use PCA-based protein concentration measurement approach. Yeah.
Fantastic. Well, Dr. Ma, I'd like to thank you again for your time today and your important research. Before we go, I would like to thank our audience for joining us today. And I'd also like to thank our sponsor, DIMA Biotechnology, for sponsoring today's webinar. Questions submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We'd also like to let you know that this webcast is available for on-demand viewing for 1 year, so that's until February 18th, 2027, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event, and please enjoy the rest of drug discovery and development. Until next time, take care, everyone. Bye-bye.
Thank you.